Hi guys, so Jason Matthew here again, and today we're going to be looking at the sodium ion channel. Now, for those of you who have been following the podcast, you would know that we have already looked at the sodium ion channel in Neuroscience 2. And in Neuroscience 2, we looked at the resting potential, action potential, refractory period, and the transmission of action potentials. But there was a lot going on in Neuroscience 2. In fact, it was over an hour long. So, as I promised, I have broken up Neuroscience into smaller parts. So today we're going to be looking at the sodium ion channel. But please, go back to the YouTube channel, BioChemGM, and look at Neuroscience 2. Because although it's long, it's important that you look at all these channels and pumps and how they work together to produce the action potential. So although I'm breaking it up in smaller pieces, I still strongly recommend that at the end of the day, you still go back to the YouTube channel BioChemGM and look at Neuroscience 2 in its entirety, because you're going to see everything coming together to produce this action potential. So you could probably use this podcast as a quick revision of a specific topic. So as I said, we're going to be looking at the voltage-gated sodium ion channel. And because this is a biochemistry course, something that you must do is you must be able to describe the structure of a sodium gated ion channel. Alright? So let's look at the structure of it. But first of all, it's a transmembrane protein. Because if you look, you see here the lipid bilayer, and you see that the protein is going across from the outside to the inside. So it's spanning the whole bilayer. So it's a transmembrane protein. Now, the voltage-gated sodium ion channel is made up of a few subunits. But the one of particular interest for us in neochemistry is the alpha subunit. So we're going to be looking at the alpha subunit. Now, the alpha subunit is made up of 4 homologous domains. So you have domain 1, domain 2, domain 3, and domain 4. So as I said, there are 4 domains. 1, 2, 3, 4 homologous domains. Alright? Now, if you look carefully, each of these 4 domains are made up of 6 helices. And we can count them. Alright, let's count them. So again, each of these 4 homologous domains are made up of 6 helices, and you can count them. 1, 2, 3, 4, 5, and 6. And if you look here, they numbered it for you, one of them. So you see 1, 2, 3, 4, 5, 6. 6 helices in each domain. Alright? Now, if you look here as well, they have color-coded some of these helices and some of these segments that join helices. And that's very important. So let's look at a few things. One, let's look at first of all, let's look at the voltage sensor. Now, the voltage sensor is color-coded blue. And if you look at each domain, there is a blue helix that is colored, well, there's a blue colored helix. And if you look at the voltage-gated sodium ion channel, that's helix 4 in each of the 4 domains. So in each of the 4 domains, the 4th helix, helix 4, is part of the voltage sensor. Now another thing that is color-coded is the activation gate. And if you look at the activation gate, you see that is helix 6 on each of the domains. So on each domain, helix 6 will make up the activation gate. Next thing is the selectivity filter, or the pore region. And the selectivity filter is made up of this segment that is in red. And you see that in each of the domain, there's a red segment. And what we say that the selectivity filter comprises of the segment that joins helix 5 to helix 6. That segment that joins helix 5 to helix 6 in each of the domain come together to form the selectivity filter. Now, so far, when we look at the activation gate, we looked at the voltage sensor, we looked at the selectivity filter. Each domain contributed to them. But for the inactivation gate, you have to be very careful. for the inactivation gate, which is in green, it's only this segment here. And this segment joins domain 3 with domain 4. It's not found in the other parts. So the inactivation gate only comprises of the segment that joins domain 3 to domain 4. So be careful about that. The voltage sensor, the activation gate, and selectivity filter, all of them, each of the domain contributes to that, except for the inactivation gate, which is the segment between domain 3 and domain 4. Alright, so what I did with this slide here was to see if you could remember what we just spoke about. So if you think that you're not ready as yet, what I strongly recommend that you do is go back, view it over again, and see if you got the hang of it, if you got all the information, and then come to this slide here. Because I want you to really apply yourself to this slide. Because if you can answer all the questions here, well then that means you know the sodium ion channel and you can move on. So what I want you to read each paragraph and write down the answers that you think correspond to the missing blocks there, or the blocks there, I should say. Alright, and as usual, I always recommend that for these podcasts that you have your pen and paper and you should be making notes along the way. Because the more you write, I believe the better you'll remember. Alright, so just a good strategy. It's something that helped me when I was going through my studies as well. Alright, so let's look at that first paragraph. Sodium channels of different tissues and organisms have a variety of subunits, but only the principal subunit. In other words, as I said before, which subunit of the sodium ion channel is important for us, especially in neurochemistry, in the development of the action potential. And you should have correctly said the alpha subunit. So you've gotten the first part correct. Let's move on. The alpha subunit is a large protein with how many homologous domains? And I know you know the answer, and you should have correctly said 4. Alright, so the alpha subunit is comprised of 4 homologous domains. So we're going good. Alright, you're ready to go ahead. Now, each of these 4 homologous domains consists of a certain number of transmembrane helices. So again, guys, if you think you're not ready for this as yet, let's go back. Go back because we have already discussed the answers for this. If you think you're confident and you're ready for it, then you should have said correctly 6. Alright, now if you think you're getting some of these things wrong, then don't go ahead. Again, go back, make sure you got the information, then move forward. Now the helix 4 in each domain, and in the previous diagram it was blue in color. Alright, so helix 4 in each domain is the voltage sensor, correct? And helix 6 is thought to be the, so you should have said the activation gate. The segments between helices 5 and 6, the pore region, form the selectivity filter. So guys, we just have one more to go. And I know you all are smart enough to realize that just by elimination, you should already know the answer. Alright? So the segment connecting domains 3 and 4 is the inactivation gate. Excellent. So let's move ahead. So in the previous diagram, we looked at the voltage gated sodium ion channel, but we looked at it spread out. We looked at the protein, from a kind of cross-section point of view. Now let's look at it from a 3-dimensional view now. The 4 domains are wrapped about a central transmembrane channel lined with, and they're actually what kind of amino acid residues you think will line the transmembrane channel. Now remember, the transmembrane channel is where the sodium ions are going to be passing through. So what kind of amino acid residues you think is going to be present there? You have 2 choices, either it's polar or non-polar. So hopefully, if you know your biochemistry, you should know that because this sodium ion channel is passing through, you would want polar amino acid residues in that transmembrane channel. Now the 4 pore regions, which was red in the other diagram, is also red here. They come together near the extracellular surface to form the selectivity filter. Now that is conserved in all sodium ion channels. In all sodium ion channels, there is a selectivity filter. Alright, now that filter is very, very specific. The filter gives the channel its ability to discriminate between sodium ions and other ions of similar size and charge. In other words, like for instance, let's look at K+, the potassium ion. Now the potassium ion is of somewhat similar size and definitely of similar charge. But this selectivity filter is so specific, it will allow Na+, sodium ions, to pass through, but it will not allow potassium ions, K+, to pass through. Alright, and lastly, the inactivation gate, which is green in this diagram here, closes soon after the activation gate opens. Alright, so I hope you all got everything there. If not, again, just keep revising. It's all about reinforcing the knowledge. And believe me, this could be kind of intimidating sometimes. So just keep practicing and you'll be good to go. Alright, so we're going on. Alright, we're looking at how this ion channel opens. So it all starts with the voltage sensing mechanism. And it involves the movement of which helix. Which helix makes up the voltage sensor. So you should have said correctly helix 4. Alright. Now it responds to a change in transmembrane potential. So what it's saying here is that let's look at this diagram here first of all. This is a neuron at rest. And you know from your previous podcast that when a neuron is in its resting potential, the outside of the neuron is positive while the inside is negative. So we say that the membrane is polarized. Alright? Now as shown as at the top, the strong, what charge is the helix here? The helix 4. So you should have said positive charge of the helix is pulled inward in response to the negative membrane potential. So remember, at rest, the inside of the membrane is negative. So the positive charge helix 4, is attracted to that negative potential on the inside. So the helix 4 is pulled inward, all right, in response to this negative membrane potential, right? So this negative charge here, and this is the neuron at rest, negative on the inside. The voltage sensor, the helix 4, is pulled inward. And when it's pulled inward, the activation gate is shut closed. It's closed, right? So in other words, no ions could pass through, no sodium ions could pass through. But then if there's a wave of depolarization, and remember in depolarization, what's going to happen is that you're going to have a change in charge. In other words, the outside is going to come negative now, and the inside is going to become positive. That's when we say the membrane is depolarized. So again, when the membrane is depolarized, so depolarization, the inside here is going to become positive. Now when that happens, there's no longer going to be that attraction. In other words, the voltage sensor helix 4 is no longer going to be pulled inward. So you see here, depolarization lessens this pull, and helix 4 relaxes by moving outward. Because when depolarization takes place, the inside is no longer negative. So there's no longer that attraction here. Alright? Because here is going to become positive when there's depolarization. So the voltage sensor is no longer pulled inward. It relaxes, it moves outward. Alright? And you see the result of that now is that this movement is communicated to the activation gate, which is orange in this diagram here. And it induces a conformational change, which is just a big way of saying the activation gate changes shape. When the activation gate undergoes this conformation change, the channel opens. In other words, this sodium ion channel opens in response to depolarization. And when the activation gate is open, sodium ions could come in. So sodium ions rush into the neuron. So let's look at this diagram here. Now they're showing you two sodium ion channels. Alright, in this diagram they show you two sodium ion channels. There's the chemically activated ion channel, and then there's the voltage gated ion channel. Now in today's podcast, we are going to focus totally on the voltage gated ion channel. That's all we have been talking about so far. In other podcasts, you will see when we do the cholinergic synapse, for instance, you will look at a chemically activated ion channel. And that's when this channel opens in response to a chemical or a ligand. In this case, you see it's acetylcholine. All right? But that is not what we are about today. Today, we are looking at sodium ion channels that are voltage gated. In other words, there must be a change in voltage for your sodium ion channel to open. So that's the focus we are looking at today. So in a voltage gated ion channel, the one that we have been looking at throughout this whole podcast, has a voltage sensor, which is this little part here. Going across now is the activation gate, and then there's the inactivation gate. And remember, at rest, this sodium ion channel is closed. So you see the activation gate is across the channel, and no sodium ions could pass through. Alright, this is when there's a resting potential. Now if the membrane becomes depolarized, alright, so it goes from a resting potential of around minus 70 millivolts. So let's say it gets less negative, it comes to like minus 55 millivolts. Alright, that the voltage sensor is going to sense that change in voltage, and the activation gate opens. When the activation gate opens, sodium ions, they come in, they rush in. So there's an influx of sodium ions. Sodium ions come into the neuron. Alright? And then there's the inactivation state, where the inactivation gate blocks the pore. So no more sodium ions come in. Now the last thing that we should talk about here is how, well, we already saw that the activation gate is activated, for want of a better word. The activation gate is turned on when there's depolarization. So you might ask the question, well, when is the inactivation gate turned on? And the thing here is this: the activation gate as well as the inactivation gate, they are both turned on when there is depolarization. So one might say, well, that sounds kind of strange, you know. Because if you're telling me that both the activation gate and the inactivation gate, they move at the same time, wouldn't that mean that the inactivation gate is going to block the ions from the get-go? But the thing is, it's not happening. So then let's use some common sense here. Let's try to figure it out. Why you think that although both activation gate as well as the inactivation gate, they are both being turned on when there's depolarization, but yet there's that time where sodium ions is able to pass through? So I hope you figured it out. It means that although they are turned on at the same time, their kinetics is different. In other words, the activation gate moves faster than the inactivation gate. So the activation gate opens or moves quicker than this inactivation gate has time to move into the pore. So it means that there will be a point in time where sodium ions could pass through because the inactivation gate hasn't reached over as yet to block the pore. Alright? So we say that there's a difference in kinetics, difference in the rate at which these gates move, with the activation gate moving faster. In other words, it opens faster than the inactivation gate could move over to block the pore. Alright? Now, all I'm saying faster and slower and stuff, we are talking about a difference of milliseconds here, but it's enough for enough sodium ions to pass through to start the action potential. So sodium ion gated, well, voltage gated sodium ion channels are responsible for initiating the action potential. So that rising part of it, and that's something that we'll discuss in another podcast. So let me see if you remembered everything I just spoke about. Activation of the voltage sensor or chemical receptor leads to the opening of a gate within the channel, which allows ions to flow through the channel. Inactivation involves a second gate moving into the channel. So although both of them are moving, the inactivation gate moves more slowly than the activation gate moves out. So that there's a time when there's no gate in the channel and ions can flow through it. So guys, that's basically sodium ion channel. Alright, and as usual, if you learned something, if you enjoyed what you heard, please show your love. Hit the like button on YouTube, as well as subscribe to the channel so you get all the videos as soon as they are uploaded. Alright? And please give us your feedback. So if you have anything to send to me, any questions, any information that you all have, please email me at jasonmatthew2011@gmail.com. I would love to hear from you guys. As usual, thank you for listening to the podcast, and thank you for the support. So take care, guys, and we'll talk more biochemistry soon enough. Take care.